last minute problem with a different link. I hope everyone will join. Thank you for joining for our second seminar of the EMSL Xchange series. Please know that we will start recording soon. It is my pleasure to host Hans Röst, currently an assistant professor at the University of Toronto in Canada. He is an expert developing computational methods for data-independent acquisition and mass spectrometry, among those the OpenSWAP software, which is now routinely used for proteomics experiments. Today, he will talk about his lab research working for now incorporating ion mobility to data-independent acquisitions workflows. Hans, please.
Okay. So welcome everybody to my talk, and thank you very much for being here. Today I will talk about improved sensitivity and duty cycle in data-independent acquisition achieved through ion mobility separation. And I would like to start out with some of the challenges that we currently have when we do bottom-up proteomics analysis. And these challenges really are due to the fact that we are analyzing very complex proteomic samples. So we are interested in analyzing samples such as whole cell lysates with 100,000 or potentially more peptides in them, analyzing complex mixtures that span across a large number of dynamic range, such as blood plasma, and analyzing highly heterogeneous samples, such as tissue samples. And so with our current LC-MS/MS methods, we can measure relatively complex samples. However, we are still limited by reproducibility, by sensitivity, and selectivity. And the reason that we are interested in addressing these problems, and the reason we're interested in addressing these problems in my lab, is that this will allow us to take the next step in proteomics and to move towards larger cohorts and multi-center studies. So if we can find a way to improve reproducibility and to make sure that proteomics mass spectrometric data is acquired reproducibly and consistently, not just on a single machine, but across multiple instruments, across multiple labs, we could do much larger studies and move into areas where we have much larger biological variability, such as human patients and human samples. Limited sensitivity is of course an issue when we are working with on the one hand with small sample amounts. So if we work with FACS sorted cells, microbiopsies, or even going down to the single cell level, we would like to improve our sensitivity as much as possible. And on the flip side of that is the question of whether we could improve the throughput by using more sensitive methods, but shortening the analysis time and therefore gaining throughput as a trade-off with the number of peptide precursors and proteins that we can analyze. And finally, if we had increased selectivity, this would allow us to separate also isobaric compounds. So starting out with the problem of reproducibility, this is really illustrated in the figure here, which is a so-called proteomics data matrix where that I've taken from a relatively recent paper, where you see in the columns a set of different samples. So these were mouse tissue samples. And then in the rows, you see individual proteins that were quantified. And one of the first things that you will notice is that the matrix is very sparse. So there are a lot of proteins we can quantify with current methods, but the quantitative data matrix is relatively sparse. And so one of the big questions in terms of reproducibility and increasing reproducibility and consistency in quantification has driven us to ask the question of how do we go from a data matrix that looks like this to a data matrix that looks like the one on the right here. And as you can appreciate, this data matrix is much more complete, has much more quantitative values, and more importantly, these quantitative values for these proteins, they are filled almost all of them are filling the complete row. So that means we now have a quantitative value for every single sample. And you can appreciate how important that is if you want to do statistical analysis or biomarker types of studies, that you have a lot of difficulty working with data where the majority, half of your data or more, is absent or missing. And so what is the difference between these two matrices? How were these acquired? And the difference here is that the matrix on the left was acquired using data-dependent shotgun proteomics, whereas the matrix on the right was acquired with a technique called data-independent acquisition, or SWAS-MS. And again, I would like to highlight that if we want to do systems-wide studies, if we want to move into the realm of large-scale clinical studies or into systems biology, we need to have systems-wide complete data, and we cannot easily deal with large amount of missing data. So for me as a computational and data science-oriented person, one of the questions was really what causes this discrepancy, and what is the difference between the way we acquire data in shotgun proteomics and in data-independent acquisition. And this basically started with the realization and observation of how a mass spectrometer samples the retention time in mass to charge space. So when we started looking at this problem for the first time, we basically looked at the problem as a sampling problem in 2 dimensions, with mass to charge in 1 dimension, and then retention time in the 2nd dimension. And so the task is now to sample at specific points in this two-dimensional space in order to produce fragment ion information. And in shotgun proteomics or data-dependent acquisition, the mass spectrometer will decide online which precursors to sample, which leads to a very positive outcome, namely that the precursors and positions that are sampled will have high intensity and likely contain a fragmentable precursor analyte. However, the downside is, because this is so tailored to one specific sample, that it really looks like you took a shotgun to your data and you have a semi-stochastic positioning of these isolation windows in this two-dimensional space. And this is really something that hurts reproducibility and is very difficult to deal with if you have different MS2 acquisitions in every single in every single sample, because it makes it very hard to compare these samples. And so the way we tried to approach this problem was saying that we, instead of using a data-dependent online algorithm to sample precursors, what we decided to do is basically divide this two-dimensional space into a grid, or into a set of pixels that would have a number of pixels in this dimension in mass to charge and a number of pixels in the retention time dimension. And we would then simply sample each of these pixels, starting with the first one in the bottom here, let's say starting with 400 to 425 mass to charge, then move on to the next pixel, sample all the analytes in that pixel, fragment them together, and produce a fragment ion spectrum of all of the analytes that were co-fragmented in that mass to charge range. So now, looking at the distribution of quadrupole isolation windows from shotgun going to DIA, this is what we what it conceptually would look like, that you simply use a relatively large quadrupole isolation window to sample this very first pixel. And for the illustration here, we have only 4 different windows. So you start down here, you sample the first quadrupole isolation window, you move to the next quadrupole isolation window, and then you move to the third quadrupole isolation window, and finally to the last window. And as soon as you hit the top, you start sampling again from the bottom. And what this really allows us to do, and the benefit this brings, is that the acquisition is discretized. We have deterministic and reproducible data acquisition. So if you shoot the same sample again in the mass spectrometer, exactly at the same positions, the quadrupole will fragment precursors and will produce a fragment ion spectrum. And so the data will look exactly the same if you perform a repeat analysis. And this is one of the main distinctions to shotgun or data-dependent acquisition. In addition, we also have this advantage that we have a complete recording of all ionizable, of all fragment ions of ionizable precursors, because with this scheme, it is not possible to miss a single precursor as long as you ensure that the cycle time, so the time between sampling one specific mass window here, 400 to 475, to the time you sample it the next time, is shorter than the elution time of a peptide. And here I've illustrated how this data actually would look like. So if you do post-processing on this data and you align all of the MS2 spectra that derive from the same precursor isolation window, you basically now get a series of spectra that are ordered in time. And if you look at specific fragment ions, what you will see is that you will see elution profiles such as this red fragment ion here across this different MS2 spectrum as the precursor elutes over the chromatographic time. And so with introducing and working on Swartz MS and data-independent acquisition, we believe that we have found a technique that really delivers this high reproducibility and this demand for reproducible data acquisition. If you have the same sample, it should produce the same data, no matter on which day you shoot it, and no matter on which instrument you perform the acquisition. However, of course, the question comes up, well, why isn't everybody simply doing this? And the main reason is that this also poses a major challenge, and mainly a computational challenge, namely that the data that is produced by this method is very complete, but it's also highly multiplexed. So we basically lose the connection of a single precursor with its fragment ion spectrum, because as you can appreciate, while we can look at individual fragment ions of a precursor, such as the red fragment ion here, we also have many other fragment ions that may come from different precursors. And this really posed a challenge in terms of data analysis. And the way we address this challenge is by treating this data as basically one big database, right? So we treated this data as if it were one large database into which we could formulate queries. And in this case, we would not use SQL to formulate queries, but we would use peptide queries to go into the data and ask very specific targeted questions, namely about the presence and absence of individual peptides. So the example that I'm taking here is, again, the series of MS2 spectra, where we see the dimension of time here. And in this case, we have 3 fragment ions. And if we perform a targeted extraction of these 3 fragment ions, what we will get is a fragment ion chromatogram that shows us how these 3 fragment ions behave over chromatographic time. And then this allows us to make a decision that we have indeed detected and quantified the peptide of interest. And obviously, we could do this manually, but we could not upscale this. So in order to upscale this approach, we wrote a software called OpenSwartz, which analyzes this whole process, performs a data extraction based on a priori knowledge. So that's using a spectral or assay library to extract thousands or hundreds of thousands of these peptide chromatograms, performs a feature detection and segmentation of the data, and finally uses a accurate null model for hypothesis testing to identify which of these peaks truly represent peptide signal and which of these peaks represent noise. Now, with this, we have addressed the problem of reproducibility of a single run and reproducibility of the data acquisition part. And what we actually realized was that now the major source of variation was the retention time. So the nano LC chromatography was now actually the major source of variation, because we have eliminated the source of semi-stochastic variation coming from the data-dependent selection algorithms used in shotgun proteomics. And so the problem that we turn to next is the alignment problem of chromatograms, where the problem basically consists in the question of which of these peaks correspond to each other, right? So you have the same set of signals, but they are shifted through squeezing and pulling in retention time, and you basically need to make a alignment across these different chromatograms to ascertain identity between all of these different peaks, and also deal with patterns where maybe you do not actually see a signal, right? Where basically the signal is below the limit of detection. And so what we set ourselves the task to do was to find and design an algorithm that would be operated on all targeted mass spec runs in conjunction and not one run at a time, as our original open source algorithm was doing, would be scalable to hundreds of runs and correct for these nonlinear chromatographic effects that we often observe in nano liquid chromatography. And only then we would really achieve this consistent and complete data matrix. And so our first crack at this problem was an algorithm called TRIC, which basically stands for Transfer of Identification, Confidence, and models the behavior of a human eye that would look at a series of chromatograms and check individual of these peaks and say, well, these are probably the peaks that correspond to each other. And we would use confidence in one of these peaks and transfer that confidence in the peak in the run to which it was aligned. And so at the time, we thought this was actually a solved problem. However, we didn't realize that we had only worked on relatively small scale studies that were done on a single instrument, on a single column, under relatively consistent and homogeneous conditions. But as soon as we started moving towards large-scale studies with 1,000 proteomic samples or more that had to be run on multiple columns or even multiple instruments, we realized that actually the algorithm that we had developed was not sufficient anymore, and specifically would not be able to work under extremely heterogeneous conditions that would introduce very large retention time errors. And so the problem that we now have picked up again relatively recently when I started my own lab, and we have designed a follow-up algorithm that is much more accurate in actually correcting for these retention time errors, because in our previous work with TRIC, we have tried to find a single global warping function that maps retention time in one run to the retention time in another run. And this was sufficient under homogeneous conditions. However, what we found is that under heterogeneous conditions, the assumption that such a global mapping function exists is actually breaking down. And so what we ended up doing was actually a much more localized approach, where we would use a technique called dynamic programming, which is a very well-established technique in computer science and bioinformatics, and we would directly operate on the raw data. So instead of trying to find one global mapping of mapping one feature to another, we would directly identify a optimal function to align the data in a chromatogram of a specific peptide in run A to the chromatogram of the same peptide in run B, and then use that localized function that could be different for every single peptide. We would compute a different warping function and use this to perform the alignment. So we have basically gone from a global alignment approach to a local alignment approach. We have gone from using features to using the raw data directly, and we still have the option to use a global constraint so that a strong local artifact can be overridden by our knowledge of what the global function should look like. So with this approach, we believe we are in a situation where we have addressed a lot of the issues around reproducibility, mainly in the areas of sampling and in the areas of retention time inconsistencies, right? And so this is now the point where we started to look into the future and think about the other two aspects that I mentioned in the very beginning, namely what are the current issues that are still apparent in SWARS-MS and data independent acquisition. And the main issue that always comes up, both from a kind of philosophical and analytical standpoint, is that whenever we do these multiplexed acquisitions, we lose the association between a precursor and the fragment ion signal. So as I've alluded to before, is with our method, we are able to extract in a targeted fashion fragment ion traces from individual peptide fragments, but these fragment ions occur in highly multiplex spectra in the background of many other peptide fragments. And this also led us to the second issue, which is an ongoing and always recurring issue in mass spectrometry-based proteomics, the limits on sensitivity. And so one of the things we realized is how these two problems are connected to the acquisition process, namely that the limited duty cycle actually means that because we have to cycle through these individual SWAS windows, and usually we use something like 32 up to 64 different windows, so that's the pixel size in the mass to charge dimension, so that if you use 32 windows and you cycle through these windows subsequently, it actually means that your mass spectrometer only spends 3% of its time on one given window. So that means of the total ion beam of a given analyte, only ever 3% of that analyte gets fragmented and then moved into the analyzer. And 97% of the time, that analyte is going into the quadrupole and gets filtered out and basically is lost for analysis. And so we reasoned, well, it's no wonder that we have issues with sensitivity if our methods inherently lose 97% of our analytes on purpose. And secondly, we were dealing with this question of intrascan dynamic range, and that is related to the fact that for any sort of analyzer, whether it's a TOF analyzer or an Orbitrap analyzer, it is very difficult to measure these very low abundant fragment ions in the presence of very high abundant fragment ions. So we are limited to about 3 to 4 orders of magnitude of intrascan dynamic range. So the dynamic range you can achieve within one single MS2 scan. And this is basically what I will talk about today in terms of novel technology development and where we believe that ion mobility can play a big role. And so what we have done is we have partnered up with several labs in Europe and with Bruker in order to develop a method called DIA-PASEF. And I'm actually very happy to announce that 2 weeks ago, the paper is now published in Nature Methods. So for anybody interested, you can now go and look that up on Nature Methods. It was already on bioRxiv since last summer. So the idea here and the insight was basically, again, looking from a somewhat data science perspective into TOF acquisition and LC separation. And one of the things we realized was that that LC separation happens on a scale of about 5 to 10 seconds on the chromatography, and we generate a spectrum on the order of about every 100 milliseconds. There is some variation on your settings, but that's more or less the scale. But what actually happens in a TOF time of flight mass spectrometer is that the true acquisition rate of spectra is on the order of 10 kilohertz. And so in order to actually acquire our spectra at 10 hertz, what the mass spectrometer really does is it takes about 1,000 spectra. So it takes the ion beam and it produces 1,000 individual TOF pushes and then averages those into one single spectrum. And so what the question here is that we asked ourselves is whether there is a better way to utilize this time instead of simply performing an average over all of this data. And the answer that we worked on here is using ion mobility or separation by collisional cross-section. And so the instrument and the device that we used here is the trapped ion mobility device that is now commercially sold by Bruker in the timsTOF Pro. And one of the very nice things to realize is that the ion mobility separation time frame very neatly fits right in between the LC separation of 5 to 10 seconds and the analysis speed of the TOF. So it's almost a perfect fit that there is a space in between here, between the 10 kilohertz of a TOF acquisition and the 0.1 hertz separation of an LC, that there is a large gap where we can very snugly fit in another separation technique that will give us additional information on the data. And so unlike a drift tube ion mobility, a trapped ion mobility device works a little bit differently, namely that here we have an electric potential illustrated here in this black line. And so the electric potential is applied in this direction, and there is a gas that flows from left to right in the forward direction. And as you can see, the electric potential is changing in the Z dimension. So that means that the ions will come to rest as soon as the force from the gas is equal to the force applied to the electric field. And so large ions will actually move further into the Z dimension than small ions. So this allows the device to accumulate ions over a certain period of time, and then in a second step, the potential barrier here can be lowered in a stepwise fashion to first release the large ion here in red, and then release the yellow ions here, and finally the green and the blue ions. And that's how you achieve separation by sequential release from a trapping device. And so this is just one example of how this data actually now looks like. So this is one single scan that usually is a single fragment ion, a single mass to charge scan, is now has a second dimension in ion mobility. And as you can appreciate, that there is a lot of complexity that is hidden here in this type of scan that you would completely miss if you projected all of the data onto the mass to charge axis, and if you did not have this additional ion mobility separation here on the bottom. So really what this adds in my view is adding a additional dimension to mass spectrometry, allowing us to separate ions in a to some degree orthogonal fashion to mass to charge and retention time. As you realize, it's not completely orthogonal because there is strong correlation structures in this data. So how do we now put these different pieces together? So we had our initial development of data-independent acquisition and the SWAS-MS targeted extraction approach, and now we have this new novel trapped ion mobility device. And so the way we put this together is we started out with our initial scheme of performing data-independent acquisition. And as you can see here, we have our individual SWAS windows that we can arrange in one after each other in order to see these elution profiles of peptide fragments. And now what happens is that each of these MS2 spectra actually gains this additional dimension. As I've shown you before in the previous spectrum, each of these MS2 spectra is actually not just a flat histogram of mass to charge and intensity, but actually has a two-dimensional, has a second dimension of ion mobility. And so what we see here is now we actually can appreciate that these three fragment ions in color here, which are assumed to be the fragment ions of a single precursor, they can be separated in ion mobility from the other fragment ions shown here in black, which are interfering fragment ions that are produced from other precursors that just happen to be in the same mass to charge window, right? So the expectation is that ion mobility allows us to separate out the fragment ions that are grouped together because they originate from different precursors that have different collisional cross-section. And this would allow us to re-establish this link between which fragment ions actually come from the same precursor and which fragment ions are background fragment ions derived from a different precursor shown in black here. So this is one of the first jobs that we thought ion mobility would do for us. The next thing that we realized was that ion mobility strongly correlates with mass to charge. So if we look at a given mass to charge window, so if we take one slice through the mass to charge domain here as shown in red here, what we realized is that actually the ions of interest, which are shown down here, the 2+ here and the 3+ ions down here, they actually do not take up the full space of ion mobility elution. So in our terms, basically we are releasing ions from the trap during a time frame of about 100 milliseconds, but the ions of interest, let's say from a given DIA window of let's say 600 to 625 mass to charge window, they would not elute throughout the full 100 milliseconds, but they would actually be confined to a relatively small part of that space. And so so we basically realized that it's relatively wasteful to put the quadrupole in isolation position and then simply allow it to measure throughout the 100 milliseconds in order to only capture these ions. So what we plan to do instead is actually allow the quadrupole to switch to different DIA windows depending on when we expect them to elute. So that in the beginning, when the large ions would elute from the ion mobility trap, we would have the quadrupole set to a isolation window that is high in mass to charge, for example, 1000 to 1025. And then once all of the 2+ ions have passed, we would switch the quadrupole to a new setting to measure ions of a lower mass to charge range, such as 800 to 825 mass to charge. And once all of these ions have passed, we would now switch the quadrupole again to select a new batch of ions, let's say 600 to 625. And then finally, we would switch the quadrupole again to capture these ions down here. So to show this in a histogram, if we look at the ion density as a function of 1/K0 here, if we simply plot a quadrupole that is set to one DIA window over the full ion mobility range, we get a histogram like this, where all of our ions appear here in the beginning, and then we have basically a lot of time that is wasted where we don't perform any measurements. Now, if we multiplex our data independent acquisition windows the way I explained on the slide before, we now perform a switch of the quadrupole. We switch to a new placement of the quadrupole here, and we can capture another population of ions from here to here, and then here we can capture a third population of ions, and finally a fourth population of ions. So this brings the DIA-PASEF acquisition scheme together, that not only is our data now two-dimensional, and every single MS2 fragment ion spectrum has the second dimension of ion mobility, but on top of that, we have the additional benefit not just of the separation here, but also the benefit of multiplexing. So instead of acquiring one single DIA window in the 100 milliseconds that it takes to perform a scan, we can now actually acquire 4 DIA windows in the same 100 milliseconds without sacrificing any acquisition time on the 2+ ions. So with this acquisition method, we now believe that we have addressed some of the main concerns that we have had around data independent acquisition, namely the loss of precursor fragment ion association. So this can be addressed by simply using the ion mobility separation aspect and separating out precursor ions, separating out individual precursor ions whose fragment ions will then fall on a different space in ion mobility. We have achieved increased duty cycle, so we actually are now 4 times faster than previous DIA acquisition methods, because our duty cycle can be so much faster, which has a lot of interesting applications, potentially also in the chromatography, where traditionally for DIA or SWAS, you always had to have relatively wide chromatographic peaks. And finally, what I haven't shown you in detail yet, but what is really intriguing, is that we also achieved increased sensitivity and dynamic range due to the ion mobility separation. And the intrascan dynamic range is relatively easy to explain, because if you do not have these black interfering ions that are co-eluting in ion mobility and basically pollute your intrascan dynamic range, you now have a much better chance of actually capturing a good signal of these colored ions here, because they are captured in their own TOF pushes and not co-captured with the other black ions here. Secondly, we also believe we have increased sensitivity because we have somewhat of a concentration effect, that instead of having these ions spread out over 100 milliseconds, they are now actually concentrated to a very narrow window of about 2 to 3 milliseconds. So all of the ions that were trapped before are now concentrated in about 10 to 20 TOF pushes instead of being spread out over 1,000 TOF pushes. Again, substantially increasing the signal to noise. However, what we then realized is that our data is now actually much more complex than the data we had before, and that we obviously needed new ways to approach data analysis. So we now had basically 5-dimensional data that had retention time, precursor mass to charge, fragment ion mass to charge, ion mobility, and intensity in its as its coordinates. And so the question then became, well, how do we deal with this 5-dimensional data? And here I'm showing you the results from one study where I'm showing you how we actually approached this problem in practice using a case study of looking at HeLa cell lysate. So the first thing that we needed to do was to create a SWAS-MS or DIA library. So we used HeLa cell lysate, fractionated it into 24 fractions, which we measured in shotgun mode. And we acquired 130,000 peptide precursors, mapping to about 9,000 unique human Swiss prot proteins or human genes. And so the library contains retention time, collisional cross-section, and fragment ion intensity for each of these 130,000 peptide precursors. And when we overlapped the acquisition windows that I've explained before, so the acquisition scheme where we would use a fourfold multiplexing. So here we have the isolation windows labeled 1 to 8, and the way to read this is that the instrument starts up here with measurement, and it measures up to this point in ion mobility, and then the quadrupole switches to its new setting down here, measures these ions, switches again, measures these ions, switches again, measures these ions. So when we look at the coverage of the peptide precursors, we actually found that we had a 97% coverage of the 2+ precursors, and 88% coverage of 3+ precursors, and an overall 92% coverage of all peptide precursors that would at least theoretically be available to analysis. The other 8% would not be available to analysis because they fall outside the measured range. The next thing we did was look at the difference in the ion chromatograms, and as expected for this peptide here, I'm showing one example, we actually saw a substantial increase in signal to noise. So this is an example of a ion chromatogram where we performed the analysis without the ion mobility enabled, and then the same chromatogram, you can even see some of the same interferences here, but much more reduced noise levels, right? So clearly going from here to here substantially decreased the amount of noise that we capture. And finally, we moved on to the data analysis. As I mentioned, this is now published just very recently, and we used two different software packages, DIA-PASEF, which we use for data conversion and library generation, and then we modified our existing open source analysis tool to handle the 5-dimensional data. And so this data analysis is now happening in a 4-dimensional space, and we are extracting chromatograms across retention time, fragment ion mass, and ion mobility here. And this really allows us to look at data in the retention time dimension, so analyze data as chromatograms, traditional retention time-based chromatograms, but it also, for each individual peptide, allows us to analyze the ion mobilogram, as well as the raw mass spectra. And for each of these different dimensions, we derive a set of scores, such as the cross correlation of the individual fragment ions in retention time, in ion mobility, and accuracy in ppm in the fragment mass space. And these set of scores are then combined using a machine learning algorithm to derive a final discriminant score. So when we look at the results of a single injection using our HeLa library on a timsTOF Pro Bruker using 200 nanogram of HeLa cell lysate on a 120-minute gradient, we actually could simulate the advances of our method relatively easily. So this allowed us to compare the original state where we started out without ion mobility, and then add these individual steps about which I've talked about, using the ion mobility, using targeted ion mobility extraction of a small window in ion mobility, and then finally the multiplexing. And so what we can see is that there's already a substantial gain from going to using the instrument as a pure TOF instrument without using any of the ion mobility capabilities to simply turning on the ion mobility device. So simply this focusing of the ions actually brought a substantial gain in identifications of about 30% increased precursor identifications, which we thought was what was actually very exciting. However, once we started to perform the targeted extraction, so not simply project all the data on the mass to charge axis, but really only extract the part of the data in the ion mobility which corresponds to the peptide of interest, we saw a very substantial 60% increase in precursor identifications. And then once we also implemented the multiplexing of fitting 4 different ion mobility DIA windows into one single scan, we saw another substantial increase in peptide identifications. So starting out from our original point of simply using a TOF instrument to our final method, we saw a almost 3-fold improvement, allowing us to identify and quantify 65,000 precursors in a single run at 1% FDR and over 7,000 Swiss prot proteins at 1% protein FDR, recovering 92% of the proteins present in the library. We also looked at quantitative performance using a synthetic mixture of human proteins and yeast, and here we saw very good performance across a large range of intensities. And finally, the last thing that I wanted to mention very quickly is that we also looked at whether DIA-PASEF would be suitable for low sample amounts. And so what we did here is basically took the same experiment as before, and we unfortunately we did not do any actual single cell experiments, but we simply took the HeLa cell lysate and diluted this using the same standard C18 liquid chromatography, and we simply went from 200 nanograms to 100 nanograms to 50 and to 10 nanograms. And we tried here two different methods. So we found that actually we could increase the sensitivity by using larger DIA windows and capturing more of the ion beam, allowing us to quantify and identify at 1% FDR over 4,000 Swiss prot proteins from only 10 nanograms of input material. With this, I would like to acknowledge all the people who have worked on this research, specifically Annie Ha and Max Frank, who have been 2 outstanding students in my lab working on this project, and our collaborators in the MAN lab, Florian Meyer, Andreas, Eugene, and of course Matthias. Our collaborators are Brooker, Stephanie, Markus, Heine, Scarlett, and Oliver, as well as our collaborators in the Abersold lab. And I would like to thank all of the funding bodies who have supported me and my lab here at the University of Toronto over the last 3 years. And of course, I would like to thank you for your attention. Thank you very much.
Thank you so much, Hans, for this great presentation and very, very informative. And now I would like to open to questions. Please, if you want to ask a question, unmute yourself and go ahead and ask. I can ask question meanwhile. Can you comment on what kind of samples specifically you think this new technology and computational workflow can give advantages that were more difficult before?
Yeah, I think that's a great question, and I think we are still at the very beginning, right? And as I have to unfortunately acknowledge is so far we most of what we have done and our collaborators have done is work on HeLa cell lysate, and so of course that's not yet one of the most exciting applications, right? And so we think one of the potentially most exciting applications could really lie in the space of where you have very low sample amounts, right? Because you so even our standard method already starts only with 200 nanograms of input protein or input peptides on the column. And so one direction where we think this could be very interesting is to go into these areas of either clinical microbiopsies where you have very limited material, or potentially sorted cells moving in the direction of potentially single cells. I think that would be very exciting. But there is a lot of other directions that we have not explored yet. So one direction we're also very interested in is post-translational modifications, where we will be, of course, interested in whether we can also combine DIA, which we have started to use for post-translational modifications, especially phosphoproteomics, whether the ion mobility separation would give us additional information, especially for phosphopeptide isomers, for example. So I think we are at the very beginning, and we have just completed the technology development, and I think the next steps are going out and identifying which applications specifically benefit. But in general, I think almost most general proteomics applications would benefit from a more reproducible and higher sensitivity approach.
Very cool. Thank you. Well, if there is no other question, I will ask one more. How difficult do you think it will be to adapt the current software that is extracting the ion mobility to process another type of data that doesn't have the quadrupole isolation? So it's more like type of the MSE, that it just have a low and high energy, and we don't have like all the precursors, basically.
Yeah, I think that would definitely be possible. We have not had a chance to work a lot with that type of data. We did some feasibility studies and in the early stages when we developed the algorithm, and were basically able to show that this is relatively straightforward for us to do. So we have adopted open source both for ion mobility as well as for a scanning quadrupole. So there is the sonar approach by Wardos, for which we have also adopted open source. So it may take a little bit of work for a specific type of data structure, but it's something that we have done in the past, and is actually relatively straightforward for us to do. And especially if it's simply the lack of quadrupole isolation, then this is quite straightforward, because we just have to tell the algorithm that there is one single isolation window in the MS2 scan that goes from 0 to 2000 mass to charge, right? So that's, I think, what we have done in the past with MS to the E. So we haven't really tried it. We have worked on MS to the E data without ion mobility, but we have not tried this algorithm on, I think it's called HD MSE, where you have ion mobility separation. So we have not tried that yet, but I think it will be relatively straightforward.
Great, thank you. Yeah, very exciting, many opportunities now. Well, with that, I think we can conclude. If anyone is interested, we will have tomorrow a series of discussions with Hans. If anyone is interested to join, please contact me. And also, if you have any questions, you can drop it in the chat and we can address it later. Thank you so much, Hans, again, and everyone for joining. Have a great rest of the day.
Thank you, everybody, and thank you for the invitation. Bye.
Thank you very much. Thank you.